Hello, I'm Dr. Shubha, Associate Professor in the Department of Biochemistry. I'm going to be handling metabolism of lipids for you all. My classes will be starting soon. So, before we start off with my session, let's look at what is there in your syllabus. You need to know about digestion and absorption, and then you need to know about metabolism of triacylglycerols, metabolism of ketone bodies, metabolism of cholesterol, uh, beta oxidation, and synthesis of fatty acids, lipoprotein metabolism, fatty liver, lipotropic factors, prostaglandins. Apart from these, you also have a few non-core topics, that is the fatty acid synthase, multi-enzyme complex, fatty acid synthesis, and lipid storage disorders. So what I'm planning to do is I will be releasing a PowerPoint with a video of the session, and I will be putting it out on Open edX for you all to go through this, and you need to prepare for that topic, probably listen to the lecture or read from the textbook, come to the class so that we can have a discussion. I will not be taking a lecture once you come to the class. So for the first topic, we are going to be looking at digestion and absorption. So let's look at what are the dietary sources of lipids. So we can have two sources, either vegetable source or animal source. So now, what are the vegetable sources that you know of? So most common sources will be the cooking oil, which we get from seeds. I think most of you will be using sunflower oil, or you might have seen your parents using coconut oil or groundnut oil. So canola oil, olive oil. So all these are examples of oils that we get from vegetable sources. So one important thing you need to know here, these fats have more of polyunsaturated fatty acids, what we call as PUFA, and they also have antioxidants, so there is less rancidity. I'm sure you would have learned about rancidity when you did chemistry of lipids. Another important point that you need to know here is that vegetable oils do not contain cholesterol, because plants don't synthesize cholesterol. Instead, they synthesize cytosterol. So whenever you look at these oils, the package, if you look at the contents, or on top clearly the manufacturer will claim 0% cholesterol. So when you see that, people will think that the manufacturer has done something to remove the cholesterol. So this brand of oil is good, other brand will be containing cholesterol. But this is just a gimmick by all manufacturers, and plant sources do not contain cholesterol. So any oil that you are using from plant source will not have cholesterol in it. Next, coming to the animal sources, we get lipids from milk, butter, ghee, that is milk and milk products, meat, fish, eggs. You might have heard of cod liver oil being very good, that is from fish. So in this also, there is a concept of visible fats and invisible fats. Visible fat is what you are adding, that you know how much you have added, how much of ghee was added or how much of oil was added. But invisible fats are those which are present in these seeds, which you do not know that, or you may not be aware that you are consuming, but it is still present in what you are eating. So how much of fats do we consume in a day, or how much should we consume? In average Indian diets, the average consumption will be around 20 to 50 grams of fats per day. And on a Western diet, it can be about 60 to 150 grams per day. But important thing to note here is that there is westernization happening everywhere. Even in the diet that we consume. So even Indian diets now consume have more fats compared to what was traditionally consumed previously. So if we look at what we are eating, what is the composition or what type of lipids are we consuming? So if we look at that, about 10% of what we eat will be cholesterol, cholesterol esters, phospholipids, and the essential fatty acids and some fat soluble vitamins. So these are just 10%, I'm telling you. So of the entire lipids that you are eating, 10% is cholesterol, cholesterol esters, phospholipids and other fatty acids. So now what is making up this rest of the 90%? So 90% of what we consume is in the form of triacylglycerols. Triacylglycerols. So one thing I want you to remember, whenever I'm using the term acyl, acyl is a common term for fatty acid. Do not confuse it with acetyl. This is acyl. So when we say acyl, it just refers to a fatty acid. So we'll be using this term again later as we move on. So I want you to remember acyl means a fatty acid. So triacylglycerol is a glycerol molecule that is esterified with 3 fatty acids, that is triacylglycerol. And before we start the actual digestion and absorption, a little bit about chemistry. I know all you have finished all these, but still a little bit about this. So we have soluble lipids and lipids which are not soluble in water. So those which are readily soluble are mainly ketone bodies and short chain fatty acids. These are the readily soluble lipids. Next we have the amphipathic lipids, that is the phospholipids. These are amphipathic, that means they have both a hydrophilic and a hydrophobic. Both these components are present. And cholesterol, free cholesterol is sparingly soluble in water because of the hydroxyl group that is present. A free hydroxyl group that is present makes it sparingly soluble. But when we look at non-polar or those which are insoluble, cholesterol esters, that is when this hydroxyl group is esterified with a fatty acid, it becomes a cholesterol ester and it is insoluble. And the triacylglycerol are also insoluble. These are the insoluble lipids. Now, we know that for carbohydrates, the storage form of energy is glycerol. Same way for lipids, the storage form of lipids is triacylglycerol, also called as triglycerides. And this is what in layman terms we call as fat depot. These are the various names for the form of lipid that we store in the body, reserve for energy purposes. And this is going to be stored in the adipose tissue. Okay, so let's look at which one is better, or when do we use these triacylglycerols. Fats are excellent for endurance events. That is when you are looking at an exercise which is going to last for a longer time, not like a 100-meter sprint, but you're looking at a marathon, something where you need continuous energy for a long period of time. That is when the body is not going to look at glycerol for energy. The body is not looking at glycogen for energy. Glycogen can only sustain for a shorter period of time. But if you're looking at something for a very long period of time, then the body will switch over to triacylglycerol or fatty acid oxidation for energy purposes. So this is commonly seen who stock up, eat for a few months, and then they hibernate for a long period of time, and they do not even uh, uh, get up for water, to drink water. So these are the hibernating animals who will be dependent on fatty acids or triacylglycerols for their energy purposes. Same thing with birds, migratory birds. They fly thousands of kilometers from one place to another place to lay eggs. So these migratory birds also are continuously using these triacylglycerol for energy purposes. Now coming to why is it that triacylglycerol is the predominant storage form? If you look at the amount of glycogen versus amount of triacylglycerol, the amount of triacylglycerol is going to be very high. The amount of triacylglycerol is very high compared to amount of glycogen. So why is it that this preference for triacylglycerol is there? So one, if you look at how much of energy we get, carbohydrates and proteins give us about 4 kilocalories per gram, whereas fatty acids give us 9 kilocalories per gram. Okay, so these are more energy rich, and this difference in calorie yield. So that is because carbohydrates are partially oxidized, so they have lot of hydroxyl groups, whereas fats are completely reduced, so you don't have hydroxyl groups at all, and it is non-polar and hydrophobic, whereas carbohydrates are polar and hydrophilic. So if the body is trying to store glycogen, then glycogen is hydrophilic and draws in water. So for every gram of glycogen that we store, it binds with 2 grams of water. So there are going to be 3 grams in that cell to store just 1 gram of glycogen. Whereas with triacylglycerol, there is no water involved. So it is completely anhydrous. If you are looking at 3 grams, it is 3 grams of pure triacylglycerol. So that way the cell is able to store more of triacylglycerol than glycogen. If we try to increase glycogen stores, then the cell is going to burst because of the water that comes in along with glycogen. So there is a limit on amount of glycogen that can be stored. Whereas with triacylglycerol, this limit is not there. So that is the reason why our adipocytes can increase, and the amount of fat content is that can be stored is very high. So you see people who have lot of fat content in their body that can be stored. So that is about a general idea about lipids. So let's start with digestion and absorption. So in this class we are going to be looking at digestion and absorption for triacylglycerols, for cholesterol and cholesterol esters, and for phospholipids. First of all, in the mouth there is no digestion happening. There is an enzyme lingual lipase, but lingual lipase is not going to be active for two reasons. One, the amount of time that the food stays in the mouth is very less. You're going to be chewing and swallowing the food. You're not going to let it stay in your mouth for an hour or so, not even for 10 minutes. Maybe within 1 minute you have chewed it and it's gone into your stomach. So there is not enough time for lipase to act, and the pH is also not so conducive for lingual lipase. So although there is an enzyme present, there is not going to be any digestion happening in the mouth. Next we we look at digestion that's happening in the stomach by gastric lipase, and then we look at what is happening in the intestine. That is the major part of the digestion and absorption. So coming to digestion of fats. Lingual lipase is secreted by the dorsal surface of tongue by the Ebner's gland in the dorsal surface of tongue. It has an optimum pH around 3 to 6.5. So that is why it is still active. It can be active when it reaches the stomach. So along with food, it comes into the stomach. And gastric lipase has an optimum pH around 4 to 6. These are secreted by the chief cells of the gastric mucosa. And these are called as the acid stable lipases, because when you come to pancreatic lipases, they require an alkaline environment, not acid. So these two are acid stable lipases, and these are acting upon mainly the highly emulsified fats that is present in milk, egg yolk, and they only act on triacyl glycerols, which are esterified with fatty acids that are either short or medium chain fatty acids. And these lipases are more specific for C3 linkage than C1. Another difference from pancreatic lipase is that these lipases do not require bile acids for their actions. So what is the use? These are used as supplements in patients who have malabsorption syndrome. That is, if pancreatic lipase is not being secreted due to any disorder, pancreatic lipase is deficient, then we can supplement the short chain and medium chain containing triacyl glycerol, fatty acids which are esterified to form triacyl glycerol. So these can be digested by the gastric and the lingual lipases. So these are used as energy supplements in patients with malabsorption syndrome. And they do not require bile salts for their activity. So these become important in neonates or infants who are on a milk-based diet, because these lipases act on highly emulsified fats. Those neonates, infants on a milk-based diet, and those individuals with pancreatic insufficiency, we give them short chain and medium chain fatty acids to make up for the pancreatic insufficiency, the malabsorption. So coming to action of these lipases, okay, as you can see here, this is a glycerol molecule esterified with fatty acid 1, fatty acid 2 and fatty acid 3. So this is a triacyl glycerol. As a first step, it is the fatty acid at carbon 3 that is going to be hydrolyzed. Okay, lipase is a hydrolase, requires water for its action. So on hydrolysis, one fatty acid is released, and now this glycerol molecule is left with two fatty acids. So it forms diacyl glycerol or 1,2-diacyl glycerol, and water and one fatty acid is released. So next, again, this lipase will act at position 1 and hydrolyze the fatty acid at position 1 and releases a free fatty acid to form monoacyl glycerol or 2-monoacyl glycerol or beta monoacyl glycerol. Now, one limitation of lipase is it cannot act at position C2. It cannot hydrolyze the fatty acid that is present at position C2. So that is why this fatty acid has to be shifted, or there is a molecular rearrangement that is required for further action of lipase, and that is possible by an isomerase. So isomerase will convert this 2-monoacyl glycerol to 1-monoacyl glycerol. So now the R2, that is the fatty acid 2, is now shifted to carbon 1, and now lipase can further hydrolyze this and release glycerol and one more free fatty acid. So free fatty acid and glycerol. So by consecutive action of lipase and isomerase, 1, 2, 3, 3 fatty acids are released and a glycerol molecule is released. So all those fatty acids which have a chain length less than 12 carbons can be directly absorbed into the intestinal cell and can be transported through blood by uh, albumin as free fatty acid bound to albumin. So this is another advantage, and that is how when there is pancreatic insufficiency we give them short chain and medium chain fatty acids because they do not require anything for their absorption. So coming to an overview, so by the action of lipase, triacylglycerol is converted to 1,2-diacylglycerol, which is further acted upon by lipase to form 2-monoacylglycerol, and then an isomerase which converts 2-monoacyl to 1-monoacylglycerol, and finally by action of lipase, glycerol is released and another free fatty acid. So what is the role of stomach in fat digestion? So mainly it helps to transfer this partially digested fat to the intestines. So thereby it is controlling the rate of delivery. To control this rate of delivery we require enterogastrone. Okay, this enterogastrone delays the rate of gastric emptying when we have eaten a high fat meal. So this is one of the reasons why we feel more full after taking a high fat meal for a longer time than if you have taken a pure carbohydrate or high carbohydrate diet. So enterogastrone is going to inhibit gastric motility and retards discharge of food bolus from stomach. So that is why fats have a high satiety value. Now coming to what happens in the intestines. Okay. So at the intestines we have the pancreatic juice that is coming in, and we also have bile that is coming in. So the passage of this acidic bolus of food into the intestine will trigger the release of pancreatic juice. And there is also the secretion of certain GI hormones like cholecystokinin, secretin and hepatocrinin. So cholecystokinin is responsible for contraction of the gallbladder and release of bile. Secretin is responsible for secreting bicarbonate. This bicarbonate is required for neutralization of this acid chyme that has entered into the intestine, and hepatocrinin helps to increase the bile formation. So pancreatic juice will contain lipolytic enzymes and it contains bile salts. Which are these lipolytic enzymes? We have pancreatic lipase and colipase which act on triacylglycerol. We have phospholipase A2 acting on phospholipids, and we have cholesterol esterase which is acting on cholesterol esters. Apart from this, the bile also contains bile salts. That is the sodium salt of glycolic acid and taurocholic acid. So how are these bile salts formed? What are they formed from? We are going to be looking at it when we discuss cholesterol metabolism. So coming to action of pancreatic lipase, this is a more powerful lipase than the acid lipases that we have seen. And action of lipase is the same as we have seen previously, that is hydrolysis and release of free fatty acids and glycerol ultimately. And this also requires an isomerase because it cannot act at position C2. Lipase can act on triacylglycerol which are esterified with fatty acids of any chain length. Whereas with acid lipases, we saw that they have a preference for only short and medium chain fatty acids, whereas lipase will act on any chain length, that is it can also be large chain or very large chain fatty acids. But pancreatic lipase also requires colipase calcium and bile salts for action. action. So what is the function of co-lipase? How is it synthesized? Co-lipase is synthesized as in a inactive zymogen form, that is as pro-co-lipase, which is acted upon by trypsin. Trypsin will hydrolyze a few amino acids, remove these amino acids and convert pro-co-lipase to lipase, co-lipase. Function is, this helps in anchoring pancreatic lipase onto the micelles that are formed later. Otherwise, pancreatic lipase cannot anchor onto the micelle and act on the triacyl glycerols. So it also prevents the action of inhibitors on pancreatic lipase. It is required in a ratio 1:1 with pancreatic lipase, and once its function is over, this co-lipase can again be used by another molecule of pancreatic lipase. Now coming to calcium. By the action of lipase, we have seen that free fatty acids are going to be released. So once there are more amount of free fatty acids that are released, this can cause a feedback inhibition and prevent further action of lipase. That is, the accumulation of these free fatty acids can inhibit the action of lipase. So to prevent this from happening, calcium is going to bind to these fatty acids and forms insoluble soaps. Calcium will bind to it and form soaps, which relieves the inhibition on pancreatic lipase. Now what about bile salts? So if you have observed at any time, if you have seen a drop of oil, if you put it in a glass of water or a container of water, you'll see that if you put multiple droplets, ultimately these droplets will all coalesce and form one big oil globule. They are not going to remain as broken up fat droplets, but they all prefer to come together and coalesce to form a big globule. So when this happens, the amount of active sites that are there for lipase will be reduced. So therefore, to prevent this from happening, we have the bile salts, which will help. So these bile salts will bind to these lipid globules and break them up into smaller globules, which are called as micelles. So these micelles will contain bile salts, cholesterol, phospholipids, and this, when it is broken down into individual globulets, then it increases the surface area that is available for lipase to act. So if pancreatic lipase by itself tries to bind to these micelles, it's not able to anchor onto the micelles. So that is where the action of co-lipase comes in, which first anchors onto micelles and helps binding of lipase and action of lipase. So now by the action of lipase, again it is going to convert the triacyl glycerol into free fatty acids and glycerol. But one thing to note here is, for conversion of 2 monoacyl glycerol to 1 monoacyl glycerol, we require isomerase. Okay, so this isomerase requires a little longer time for its action. So that is why, if we look at what are the contents in the intestinal lumen, we are going to see glycerol, free fatty acids, and a large amount of 2 monoacyl glycerol, and maybe about 6%, around 6% will be 1 monoacyl glycerol. So coming to digestion of cholesterol esters. So free cholesterol generally has a hydroxyl group, and this hydroxyl group will get esterified to a fatty acid to form cholesterol esters. Okay, so we need an enzyme called cholesterol esterase which will act on this, which will hydrolyze the cholesterol ester and release a free fatty acid and cholesterol. So now the intestinal lumen is going to have free fatty acid and cholesterol as well. Coming to phospholipids. Okay, phospholipids, there is an enzyme called phospholipase A2 which hydrolyzes and releases the fatty acid that is present at position 2. So we have a fatty acid that is released and the phospholipid is now converted to a lysophospholipid. And this lysophospholipid that is present is amphipathic in nature and therefore helps in micelle formation and helps in absorption of these digested lipids. So ultimately, the intestinal lumen will contain phospholipids, lysophospholipids, cholesterol, glycerol, free fatty acids, 2 monoacyl glycerol and 1 monoacyl glycerol. Coming to absorption, so these mixed micelles will contain all these, and by diffusion they are able to transport the lysophospholipid, cholesterol, free fatty acids and the monoacyl glycerols into the epithelial cell in the intestine. Okay, now within the cell, in the intestinal cell, the glycerol that is absorbed and short chain fatty acids or less than 12 carbon length fatty acids can be directly absorbed into the bloodstream. They do not require any carriers, or the small chain fatty acids will bind to albumin and free fatty acid and albumin complex will be transported. Glycerol does not require any transporter. Whereas the 2 monoacyl glycerol, 1 monoacyl glycerol and the free fatty acids that are there are being utilized and triacyl glycerol will be resynthesized. The cholesterol that is present is again reesterified with the fatty acid to form cholesterol ester. And the intestinal cell will synthesize apolipoproteins which are used for formation of chylomicrons. Chylomicron is a lipoprotein. We will be looking more about chylomicron in the class when we are discussing about lipoproteins. But right now you need to know that chylomicrons are synthesized by the intestines and these are mainly for transport of triacyl glycerol and the cholesterol esters. So these chylomicrons are released into the lacteals and through these lymphatic supply, ultimately they reach the blood and then they are transported across to all tissues where the tissues which require fatty acids will take up these free fatty acids and utilize them. So there is an enzyme lipoprotein lipase present in the capillary walls which will act on the triacyl glycerol to release free fatty acids. So usually if we collect a sample of blood after maybe 2 hours after a high fat meal, you can notice that the serum will be more milky because of the presence of these chylomicrons. And by the action of lipoprotein lipase, which also requires heparin for its action, this amount of chylomicrons will be reduced and therefore the milkiness that is there in the serum is also decreased. So that is why heparin is called as the clearing factor. It takes about 5 to 8 hours for complete disappearance of these ingested chylomicrons that are transporting the ingested lipids. So a small note, in case you are getting a question about what are the physiologically important lipases, we have discussed about lingual lipase and gastric lipase. We have discussed about pancreatic lipase, And I have even mentioned about lipoprotein lipase, although we'll look at it when we come to class on lipoproteins. I have told you about phospholipase A2. Apart from that, there is one hormone sensitive lipase which is present in adipocytes. So we look at this when we need to discuss about beta oxidation. So there is a hormone sensitive lipase. So these are the various different lipases that are present in the body. Now coming to disorders, what are the disorders that are associated with lipid digestion and absorption? So malabsorption of lipids leads to excretion of fat in feces. So when this is more than 6 gram per day, it is called as steatorrhea. More than 6 gram per day of fat in feces is called steatorrhea. So either there is a defect in digestion or there is a defect in absorption. So there could be an impairment in breakdown of fat. That is, the pancreatic juice and the bile juice is not reaching the intestine. Or there is a defective micelle formation. Or the cell itself is defective and is unable to absorb. Or the cell has absorbed but it's unable to synthesize chylomicron and transport this absorbed lipids into the bloodstream. So these are the various potential problems that we can have at different levels. So if we are looking at defective digestion, either there is pancreatic insufficiency because of which lipase is not available, or there is obstructive jaundice because of which the bile juice and the pancreatic juice is not reaching the intestines. Or there can be chronic pancreatitis leading to pancreas unable to synthesize lipase, or there is an inherent or inborn defect in the lipase or colipase itself. Coming to defective absorption, there are various causes. It can be due to celiac disease, Sprue, Crohn's disease, or maybe due to some strictures or some other cause, or due to some cancers. There is surgical removal of large parts of the intestine so that there is not much time available for action of lipases, and there is defective absorption of these lipases. And defective absorption can also be due to obstructive jaundice, as bile salts are required. So I guess with this we come to an end of digestion and absorption. So if you have any queries, we shall be discussing this in the class. So before I finish, there is a small activity that I want you all to attempt, and we can discuss the answers in class. I'll also put up one of this, I'll put up this slide as a PDF so that you are able to look only at this crossword and attempt it. And for further reading, that is, what do I want you to read next or read more when you are coming to the class? I want you to look at what are the therapeutic uses of these enzymes, or in which group of patients are we using these enzymes as medicines. So in enzyme class you have learned that there is a therapeutic use of enzymes also. So what are the therapeutic uses of these enzymes? And what is the application of the knowledge that you have learned now in designing drugs for reducing obesity? So where are we making use of this knowledge of digestion and absorption of lipids? So that in pharmacology you will come across what drugs are available to reduce absorption and help in management of obesity. So these are two things I want you to read further and then come to the class. If you have any doubts, we are going to be discussing it in class. Thank you.